In this section, I'm going to talk about internal eukaryotic cell structures. So starting with the nucleus is sometimes known as the control center. It's a sphere that's most prominent in the organelle, or in the most prominent organelle in the cell, It's separated from the cytoplasm by the nuclear envelope. The nuclear envelope is simply the boundary of the nucleus. Nuclear pores are going to be holes in the nuclear envelope, and they allow for substances to move into and out of the nucleus. The nucleolus stains more intensely due to its RNA content, so it's the dense part in the center of the nucleus, and this is the site for ribosomal RNA synthesis. So your ribosomes are made up of some protein and then ribosomal RNA, or rRNA, and they're made in the nucleolus. The genetic material of a eukaryotic cell is linear. So the chromosome structure is linear, L-I-N-E-A-R, like a line. So they're like little sticks of chromosomes. Eukaryotic organisms can be haploid or diploid. Haploid means that they only have one copy of each chromosome. Diploid means that they have two copies of each chromosome. Now, humans are predominantly diploid, with the exception of the egg and the sperm, which are haploid. And a lot of organisms go through similar types of life cycles, which we're going to talk about some fungi and some eukaryotic parasites later on in this lecture. Chromatin and histones. So chromatin is condensed DNA. It contains DNA that's wound around these histone proteins. Histones are proteins that help wind up and condense the DNA. So your DNA is basically a long strand of stuff. In order to get it all condensed and compacted, we have these proteins. And the proteins, histone proteins, they're like little circular proteins. So our strand of DNA can be wound around these proteins in order to make it more condensed. Think about when you wind up a cord, like let's say the cord to your laptop or your cord to plug in your phone or something like that. Now, if you just roll it in a ball and throw the cord, that cord's going to unravel and it's not going to stay compacted. But if you wind it around something, like maybe the plug part of the cord, if you wind it around that, it's going to stay condensed. And that's how we condense our DNA. The DNA material is wound around histone proteins, and together, this is called chromatin. Replication. So eukaryotic organisms can replicate or make copies of themselves through a couple different processes. Budding. Budding is when you have one large cell and then little small copies kind of break off of the large cell. Budding is common in yeast. Mitosis is how our cells divide. So mitosis, you have a cell, a large cell, that splits in half to make two smaller cells. In meiosis, you have a larger diploid cell that goes through 2 rounds of division to eventually create 4 haploid cells. And that's how we produce our gametes, our sperm and our egg, but other organisms, microorganisms that are eukaryotes, also reproduce in that way. The eukaryotic ribosome. So when we compare eukaryotes to prokaryotes, eukaryotes have a larger ribosome. You don't need to know the specific sizes of each one of the subunits or anything like that. You just need to know that the eukaryotic ribosome is larger. Ribosomes are responsible for protein production, and there's a big subunit and a little subunit. Endoplasmic reticulum. Surrounding the nucleus is endoplasmic reticulum, and there's smooth ER and rough ER. Smooth ER is responsible for lipid metabolism and detoxification of the cell, while rough ER contains ribosomes, and that's responsible for protein synthesis. Rough endoplasmic reticulum will be responsible for producing proteins that are shipped out of the cell. So they're either going to be embedded in the cell membrane or in the cell wall, or shipped out of the cell entirely. There's an organism that you've probably heard of called chlamydia. Chlamydia trachomatis causes severe chlamydia infections. Chlamydia is a unique bacterium in that it has to go inside of a cell in order to infect your cells. So like other types of infections, like if you get a staph infection, it's just on the surface of your skin, it stays on the surface of your cells. If you get like athlete's foot, that's again on the surface of your skin. But chlamydia is an intracellular parasite or an intracellular pathogen. Intra means inside of, cellular is talking about inside of the cell. So what chlamydia does, and it's kind of interesting how it does this, is when chlamydia gets inside of your cells, it converts from this really dormant, solid, tough form to this loose form. And this loose form produces proteins that can modify how your cell acts. So if we're talking about a vaginal infection, chlamydia gets near vaginal cells. The chlamydia organism will enter inside of the cells of the vagina, and then it will go near to the nucleus. And when it gets close to the nucleus, what it does is it actually takes over the production in the endoplasmic reticulum. Early on, it's going to cause the endoplasmic reticulum to have a lot of ribosomes on it. So ribosomes are going to stick to this endoplasmic reticulum, and it's going to force your cells to make chlamydia proteins. Later in the life cycle, chlamydia is done taking over your cell, and now it wants to leave your cell to go infect the next cell. So it's going to need some lipids, some fats in order to do that. So it shifts production. It tells these ribosomes to leave your endoplasmic reticulum, converting your endoplasmic reticulum into smooth ER. And now it forces your endoplasmic reticulum to produce lipids for it so that it can have its own membrane and escape your cell. So it's kind of cool how chlamydia has the ability to moderate how our endoplasmic reticulum works. Just because we're seeing rough ER studded with ribosomes now doesn't mean that those ribosomes are stuck forever. Ribosomes will fall off and stick depending on the needs of the cell. So in the case of chlamydia, it forces your endoplasmic reticulum to do this. But if we're talking about just a generalized human cell, if you need to make hormones, for example, in the cell, all these ribosomes are going to fall off, and you're going to be able to produce lipid hormones in the smooth endoplasmic reticulum. If the cell suddenly needs more protein, it's going to tell the ribosomes to stick, and the endoplasmic reticulum is going to be converted to a protein-producing factory. So this isn't stuck how it is. These ribosomes can fall off and stick depending on the need of the cell. The Golgi apparatus packages and modifies substances to be shipped out of the cell. It consists of a stack of flattened disc-shaped sacs. It's closely connected to the endoplasmic reticulum, and there's kind of a conveyor system that transports stuff. The information from the nucleus is then produced into a protein in the endoplasmic reticulum, and then it's going to be packaged into a vesicle and transport to the Golgi apparatus. The Golgi apparatus is going to modify it, repackage it into another vesicle, and get it for ready for shipment to the outside of the cell. So here you can see a picture of the vesicles, and the vesicles can fuse with the Golgi apparatus. So we start here in the endoplasmic reticulum, proteins are produced here. From here, vesicles are going to transport the substances to the Golgi apparatus, and then those substances are going to be modified in here, and they're going to be repackaged into vesicles and eventually escape to the cell surface. The nucleus, endoplasmic reticulum, and Golgi apparatus are nature's assembly line. Genetic information originates from the nucleus. Proteins are synthesized on the ribosomes and deposited into the rough endoplasmic reticulum. And then proteins are transported to the Golgi apparatus to be modified and packaged into vesicles. So I like to give this analogy every time I talk about protein production. So let's say you wanted to bake a cake for your aunt in Arizona. I actually have an aunt that used to live in Arizona, so this used to make a lot of sense for me, but just stick with me for this analogy. So we start with the cookbook. And we can compare the cookbook to your DNA. So the cookbook contains all of the recipes for anything that you could possibly make. Your DNA contains all of the recipes for anything that you could possibly make. Now, from this cookbook, we're going to read or flip through the pages, and we're going to make a copy of a certain recipe. So we're going to copy down a recipe. And that recipe is similar to a gene or a messenger RNA copy. And if you've never heard of messenger RNA before, we will circle back to this exact same analogy when I talk about genetics, and we'll talk more about this recipe part and how the recipe is made and turned into a protein. So from that recipe, you, or a ribosome, are going to make a cake, or your ribosome is going to make a protein in the kitchen, or in the rough endoplasmic reticulum. Once your cake is baked in the kitchen, you're going to get into your car, or the protein is going to get into a vesicle, and you're going to drive to the post office. Or the Golgi apparatus. The post office is going to package up your cake, put it in a box, put an address label on it, get it ready for shipment to your aunt in Arizona. Similar to what the Golgi apparatus is going to do. The Golgi apparatus is going to package and modify that protein so that it's ready to be activated once it hits the cell surface. From here, it's going to get into a mail truck, or another vesicle, to be delivered to your aunt in Arizona. In this case, we would deliver it to the cell surface. So I will circle back to this analogy, like I said, when we get to talking about transcription and translation, protein production in the genetics lectures. Cellular vesicles, lysosome. To lyse means to cut up or break apart. And that stem is going to come back quite a bit in this course. So knowing that now is going to help you. So lysosome, lyse means to break apart. Lysosome contains digestive enzymes, and that's going to help dissolve or digest substances that were brought into the cell. I sometimes refer to them as the mini stomachs of the cell. Vacuoles. Vacuoles are membrane-bound sacs that contain fluids or solid particles to be digested, excreted, or stores. So vacuoles can sometimes store nutrients for the cell. If you think about that single-celled organism that's floating around in a lake somewhere, and that single-celled organism needs nutrients. So in a lake, there's not that many nutrients that are condensed. So what that organism is going to do is it's going to, when it does find a food source, take in all of those nutrients and store them in the cell. That way, if they don't come across another nutrient source for a couple days, they're good because they have those nutrients stored inside of there. But vacuoles can contain all sorts of different things depending on the needs of the cell. The mitochondria supplies the bulk of the energy to the cell. So mitochondria are the predominant sites for energy production. Small amounts of energy is produced in the cytosol of the cell, or the background of the cell outside of the mitochondria. But a majority of the energy is produced in the mitochondria. Mitochondria have cristae, which are folds on the inner membrane that help enzymes and electron carriers of aerobic respiration. And then the matrix holds up the ribosomes, DNA, and enzymes and other compounds used in metabolism. As I mentioned previously, and I talked about this endosymbiont theory or endosymbiosis, I said that there was some pieces of evidence where this theory came from. So some unique characteristics of mitochondria that support the endosymbiont theory is that they divide independently of the cell, they contain circular DNA, and they have bacterial-sized 70S ribosomes, so smaller bacterial-sized ribosomes. They also have those two membranes. So I talked about the inner membrane and the outer membrane, and that's all evidence toward that endosymbiont theory. Chloroplasts are the center of photosynthesis. Eukaryotic cells that have chloroplasts include algae and plants. They are capable of converting energy into food. So basically in photosynthesis, you take energy from the sun and you end up with a food product. So they create their own sugar. That sugar can then be broken down and turned into energy through the mitochondria. And the cell ends up getting energy, more energy by breaking down the food than it costs to make the food in the first place. So there's a net gain of energy overall. Photosynthetic role of chloroplasts, primary producer of all organic nutrients and primary producers of oxygen gas. And then cells have a cytoskeleton. So the functions of the cytoskeleton include anchoring organelles, moving RNA and vesicles around, and permitting the shape changes and movement of the cell. There's three main types of cytoskeleton filaments. Actin filaments are the thinnest. Intermediate filaments are intermediate in size. Intermediate filaments provide more of the structural support of the cell and cell shape. So intermediate filaments are very similar to like a human skeleton. So they're going to make up the bulk of the structural support in that cell and give the cell its shape. While actin filaments are more for allowing the cell to move. Microtubules are large tube-like structures that can grow and shrink to move large substances around inside of the cell. Microtubules also make up the basis of flagella and cilia. And then this is just a chart that you can review of which organisms have which organelles.